All right, so we covered the bones. Now we transition to muscle. So really bones and muscle make the bread and butter of our class. So you really want to spend time with the next few chapters really understanding muscles. But before we start memorizing a whole bunch of origin, insertion, and these muscles, we have to understand how muscles work, what makes up muscle. So athletes rely on toned skeletal muscles to supply the force required for movement. All right. Now there's a difference. There's three different types of muscle, and we're talking about skeletal, cardiac, and smooth. So here are the three types: skeletal, cardiac, and smooth. The muscular system is just skeletal muscle, and myology is the study of skeletal muscles. What's the functions of muscle? Well, they provide movement of body parts, body contents, they provide communication, they provide stability, so maintain posture, resist pull of gravity, they control body openings and passageways, mouth, anus, they provide heat, so heat production necessary for enzymes to function, and they have glycemic control, so they absorb, store, and use glucose. What are the properties of muscle? So this is what muscles can do. Well, they have excitability, responsiveness, react to stimuli. They have conductivity, they can spread electrical impulse through muscle cells. They can contract, or contractility, they shorten when stimulated. Extensibility can stretch without harm. And elasticity can recoil from stretch. So when you look at from sports or athlete perspective, the ability to have extensibility and the ability to have elasticity, I think, are two key components to preventing injury. If you can't stretch without harm, you're going to go beyond it and you probably get some tears. And if you can't go back to its original state, can't recoil from the stretch, then you don't have any push-off and you don't have any power. Okay, so I really think that extensibility and elasticity play a key role. And as you get older, unfortunately, your extensibility and elasticity, or after injury, these are affected. Now, let's look at skeletal muscle. That's voluntary. That means you decide when you want to move. You decide when you want to flex those biceps at the beach. They're striated. They have alternating light and dark bands. They attach to bone. And they have muscle cells, which are muscle fiber or a myofiber. Now, cardiac is striated, so striated, but it's involuntary. Again, you don't have to tell your heart when to beat. It is under automatic control. The cell is the cardiomyocyte. Okay. And then you have smooth muscle in your intestines and your GI tract. Involuntary, again, you don't have to tell your stomach or your small intestine to digest. It'll do it, but it's non-striated. Okay. It has a fusiform cell shape and it's tapered at the ends. So make sure you know the characteristics of each one and what they look like. So here are the three types. A is skeletal muscle, B is smooth, and C is cardiac. So if you look at skeletal muscle fibers, they're going to have nucleus, they're going to have the muscle fiber here, they're going to have an endomyosin, and they have striations. So you should be able to recognize a skeletal muscle as a histology. Okay, and we'll talk about what an endomyosin, perimyosin are in just a second. Those are layers. The ultrastructure of muscle fibers. Now this, you're like, oh my gosh, I didn't think that muscle fibers were that complicated, but they are. They are. You have a sarcolemma, which are transverse tubules. So here's the muscle fiber itself. So if you were to break down the muscle fiber, then it's all these little things that are embedded in the muscle fiber. Of course, you have nucleuses, you have myofibrils, you have glycogen, you have myoglobulins, and you have a sarcoplasmic reticulum. Okay, and these all play a role in the ability to contract, and we'll talk about the physiology of it in just a second. But just understand the parts of the muscle fiber first. Okay, nucleus, sarcolemmas, myofibrils, okay, sarcoplasm, sarcoplasmic reticulum. Now, the sarcomere, okay, the sarcomere is the functional unit of skeletal muscle fiber. That's a great quiz question. So the sarcomere is the region from one Z line to the next. Boom, boom. So that constitutes the functional unit of the skeletal muscle fiber. Okay, and we'll talk about the Z line, the M line, the H line. But I want you to understand that this is a great quiz question, that the functional unit of skeletal muscle fiber is a sarcomere. And that's measured from one Z line to another Z line. Okay. Now, you have thick filaments and then you have thin filaments. And we'll talk about those as well. But how do muscles really work? It's important to understand the mechanism of muscle contraction and relaxation properly before learning the difference between troponin and tropomyosin. And that's important as well. So muscle fibers themselves are composed of myofibrils. Myofibrils are made up of long proteins arranged in sections called sarcomeres, which are the functional unit or basic building blocks of striated muscle tissue. So this is all terminology that you need to know. Then, sarcomeres has two components called a thin and thick filaments made up of actin and myosin proteins. Thick and thin filaments of myosin and actin are arranged next to each other within the sarcomere. The interaction of these two proteins within each sarcomere causes the sarcomere to shorten, which in turn causes a muscle contraction. Okay, so it's the sliding filament theory. They slide. During sarcomere contraction, myosin heads in the thick filaments bind with actin in the thin filaments and pull the thin filaments towards each other. The ends of the sarcomeres are pulled closer together, shortening the length of the muscle fiber. So see, it's actually pretty complicated how you just contract one muscle fiber, but it's amazing how it does it. Now, calcium ions are required for the contraction of the sarcomere, so this is where nutrition plays a role in having the right amount of ions, sodium, calcium. When calcium ion concentration rises, the muscle contracts, and when it is low, the muscle relaxes. Troponin and tropomyosin are two proteins which regulate sarcomere contraction via calcium binding. So when calcium ions are present, calcium binds with troponin and removes the tropomyosin. Okay? It exposes the myosin binding site in actin. When the muscle is relaxed, tropomyosin blocks the myosin binding sites in the actin filaments. And I'll show you a video of this so it makes sense. But the key difference between troponin and tropomyosin is that troponin frees the myosin binding sites of actin filaments, while tropomyosin blocks the binding site. So these are all great quiz questions. Now, so in order to understand this, well, let's look at a heart attack. So an elevated troponin is in general concerning for heart damage until proven otherwise. So it can be from a heart attack due to a blockage or due to other conditions that place strain on the heart, such as heart failure or critical illness. So non-cardiac causes of troponin elevation could be serious illness, pneumonia, kidney, and disease. So if a patient comes into the ER and they say they have chest pain and they've had it just recently, and it's like an elephant on their chest, and then they do a blood work and their troponin levels are high, then they can say, okay, they probably had a heart attack. So, and then they can do an EKG as well. So those are some things. There's really 3 ways that they can see if a person had a heart attack. Now, someone that's younger that comes into the ER, they might have chest pain, but that could be anxiety, that could be stress, it could be a lot of things. They could be extracurricular drugs that they took, but it might not be a heart attack. So there's other checks and balances that they can do to check if it's a heart attack. And one of the key levels is checking troponin levels in your blood to see if you've had a heart attack. Good quiz question. All right, so motor end plate and innervation at the neuromuscular junction. The axon terminal releases acetylcholine. The motor end plate is the location of the acetylcholine receptors in the muscle fiber sarcolemma. When acetylcholine molecules are released, They diffuse across a minute space or a minute space called the synaptic cleft and bind to the receptors. Okay, so that was from the video. Again, sarcolemma, sarcoplasmic reticulum, T-tubule, and the triad. Contraction of muscle fibers, and action potential arrives at the neuromuscular junction. So what you want to do is, this is the basis of brain, muscle, and heart. Right? So we're going to talk a lot about this neuromuscular junction and how neurotransmitters are released and how it affects it. So you want to pay attention to how this works. So an action potential arrives at the neuromuscular junction. Acetylcholine is released, binds to receptors, and opens sodium ion channels, leading to an action potential in the sarcolemma. Action potential travels along the T-tubules. Okay. The thin and thick filament interaction leads to muscle contraction. Muscle shortens and produces tension. So all that has to occur just for you to flex your biceps. Now, how do you relax? Calcium ions are pumped back into the sarcoplasmic reticulum, which causes the tropomyosin to reshield the binding sites on the actin strands. A muscle may also stop contracting when it runs out of ATP and becomes fatigued. So no mitochondria, no ATP, then it becomes tired, and that's why you can get cramps, or you can just totally relax. Okay, so muscle relaxers can work this way. Now again, certain drugs, and when we talk about the brain, we'll talk about more neurotransmitters and how they work and how they can mimic certain neurotransmitters. But really how muscle relaxers can work is that obviously you know that we need acetylcholine to arrive at the neuromuscular junction, and it has to be released. So certain muscle relaxers will mimic acetylcholine and they will prevent it from happening. Again, it's more complex than that, but at least you understand that certain drugs, even recreational drugs, will mimic certain things. So there's certain drugs that will mimic dopamine, serotonin, right? So that's why you feel good when you take certain drugs, because they're mimicking a natural neurotransmitter that occurs in your body. Okay. So again, when we do the brain, I'll tell you all the neurotransmitters that are normal in your brain, but then what recreational drugs mimic, and that's why you get the high that you do. Okay, so good stuff. The sliding filament model of muscle contraction. When a sarcomere contracts, the Z lines move closer together. Okay, and the I band becomes smaller, so these move closer together. Skeletal muscle contraction, the active site on actin is exposed as calcium binds to troponin. The myosin head is attracted to actin, and the myosin binds actin to its actin binding site, forming the cross bridge. During the power stroke, and again, that video was awesome, the phosphate generated in the previous contraction is released. This results in the myosin head pivoting toward the center of the sarcomere, after which the attached ADP and phosphate group are released. A new molecule of ATP attaches to the myosin head, causing the cross bridge to detach. The myosin head dehydrolyzes ATP to ADP and phosphate, which returns the myosin to the cocked position. So yes, that's how complicated muscle contraction is. Really not complicated, but there's a lot of terminology that probably confuses you, so just watch that video over and over again. Okay. Now, again, just review. This is where you probably want to highlight, and this is what you study. So you have thick filaments called myosin. Their heads project from the bundle tails of hundreds of molecules. Then you have thin filaments called actin. So you have a fibrous actin that resembles the necklace. You have a globular actin that resembles one bead of the necklace. The active site, the myosin binding area on the G-actin. Now you have tropomyosin, which blocks active sites when the muscle is relaxed. And then you have troponin, which attaches to tropomyosin, binds calcium when excited. So both of these need to work in order to expose and cause muscle contractions. Again, the elastic filaments. Here's the head, the tail of the myosin. The tighten anchors thick myofilaments. Functions contractile proteins are myosin and actin, and the regulatory proteins are tropomyosin and troponin. Okay. When you have a heart attack and you have damage, then you might have an increase in troponin in your blood. Don't worry, be happy now. Take 17 muscles to smile and 43 to frown. Unless you're trying to give your face a bit of workout, smiling is a much easier option for most of us. Anyone who's ever scowled, squinted, or frowned for a long period of time knows how it tires out the face, which doesn't do a thing to improve your mood. And as you know, you might have friends or you have colleagues or coworkers that have RBF. And those that know RBF, see, Patel knows some slang, and you're just thinking, why? Why are you like that? Why is that? Life isn't that bad. Get rid of that RBF. Okay, and then there's RDF as well. Guys can have that as well, but we see RBF quite a bit. Okay, so let's don't worry, be happy now. All right, striations, narrow stripes of light and dark that run perpendicular to the length of the cell. You have the A band, which is a dark stripe of thick myofilaments that partly overlap thin myofilaments. You have the H band, central region of A band, that is lighter due to the lack of thin myofilaments. I band, which is a light stripe of thin myofilaments only. You have the Z disc, midline of I band. Okay, so these are things, but I really think that video will help, and I can show that video over and over again to really understand how muscles contract. Okay, so here it is again. A sarcomere is from Z to Z, and then here is the M line, and then here's the thick and thin filaments, and they pull on each other to cause a muscle contraction. Blood supply to the muscles. At rest, muscle receives about 1/4, 25% of the heart's output. During heavy exercise, though, muscular system receives more than 75% of the heart's output, a larger percentage of a larger output. Muscle is generally well vascularized. So from a healing standpoint, after bone, muscle has the best blood supply. So really a muscle strain should heal rather quickly. Now, again, terminology, people use sprain versus strain, and it's not the same. So you strain, ST, strain your muscles, but you SP, sprain ligaments. Make sure you know the difference between a sprain and a strain. Okay, the nerve-muscle relationship, again, the neuromuscular junction, synapse, motor neuron synaptic knob contains vesicles with acetylcholine. Acetylcholine we're going to use in the heart, and we're going to use acetylcholine in the brain as well. You have the synaptic cleft, postsynaptic membrane, folds of muscle cells, membrane containing acetylcholine receptors. Okay. And then you have a motor unit, consists of one motor neuron and multiple muscle fibers. Okay, behaves as a single functional unit, meaning it's all or none. Fibers from one unit are dispersed in the muscle, and so it's either going to fire all these or none of these. So it's called the all or none philosophy, and that's the motor unit right here. And so certain, let's say your eye and certain hand muscles have not too many motor units, whereas other muscles can have a lot of motor units. Now, again, how do you contract and how do you relax? Excitation, a nerve signal arrives at the knob. Okay, the knob releases acetylcholine, which binds to the muscle's receptors, opening ion gates and exciting the muscle. Excitation spreads down and through the muscle cell. So you need acetylcholine first to excite. Then the electrical impulse triggers calcium release from sarcoplasmic reticulum. So acetylcholine, then calcium release. Then the calcium must bind to troponin, which moves the tropomyosin to expose the actin. So this guy is like the gatekeeper. Say, hey, get out of the way, tropomyosin. And in order to move the tropomyosin, we need troponin. Okay? I know it's a little complicated, a lot of words, but it's actually relatively simple. Then myosin breaks down ATP and extends its head. Myosin forms a cross bridge with actin, and myosin pulls the actin in a power stroke, then detaches and reattaches. So it's like rowing, right? You guys go rowing, it goes, imagine the head going into the water, boom, attaching, and then moving forward. And then relaxation, well, the nerve signal stops, sarcoplasmic reticulum reabsorbs the calcium, the troponin blockade resumes, and tension subsides. Excite, relax, excite, relax. 10 real steps into just a simple muscle contraction.